Hello, just in time. No new handouts. It's all the same stuff from last time. But you're just in time to get into new stuff for the slides. I don't think there's very many things I need to draw, but not very many things, but I'm sure I'll have to erase these to-do lists at some point. So just real quick, on the left-hand side, if we tackle that, today we go through pregnancy, growth, and development, as well as a little bit of a heredity PowerPoint. Thursday, we just have review for exam 5, as well as I'll set up that practice practical. A week from today, we have our exam and practical for unit number 5. The day after that, this Thursday, technically dead day, there's no official class. If you don't show up, you won't miss any new information. However, it's my only day to return exam 5 to you at a day that we can go over exam 5. You can pick up exam 5 the day of the final exam, but I just won't be able to answer a lot of questions about it because we'll be in a testing environment. So feel free to show up to this review day if you want to get your exam back and go through additional stuff before the final exam. I tried to put the notes that the final exam is all multiple choice questions. I would recommend reviewing those previous class handouts from each of the units. I know that's a lot. It's kind of hard to do all maybe the night before the final exam, but if you've been keeping up with those class handouts throughout the semester, then you should be ready to go for that final exam. Practice practical, we said, were things that have shown back up on previous practicals. And welcome, you're just in time. We are about to start the slides. Those are the same handouts from last time. You probably got those, and I think you got your exam and practical back. So remember on D2L, there are quizzes 9 and 10 that are male and female reproductive stuff. I think those are still there today, but they may go away.
One goes away tonight, one goes away Thursday.
Male goes away tonight, female goes away Thursday night. Then the stuff we talk about today is really just on the bonus quiz. So it makes up about 10 to 15% of the stuff on exam 5. What else? Over here on the right-hand side, I started with a to-do list of the things that we need to point out for our pregnancy, growth, and development PowerPoint, as well as I started with a list on the key items from the heredity PowerPoint, but I wanted to leave myself a little bit of room to write some things out. So at some point, I'm sure we're going to erase that and draw some things on the board. But looking more closely at our to-do list for the pregnancy, growth, and development, what we really are going to get into here, just to kind of break this down, in our previous 2 lectures, we're looking at details of male and female reproductive system. And one of the key things that we were focusing on is how each system had its own strategy for developing these gametes, these haploid cells that have only one set of chromosomes, so they're not like normal body cells, and the immune system wants to take them out. So I think we did sketches of ovarian and uterine cycle, and then we did a sketch of the cross section through a seminiferous tubule, and it would be, I'm sure, good to remember the cells and the seminiferous tubule and the stages of the ovarian and uterine cycle. But anyway, that's review from last time, development of these gametes. Today where we're going is we're going to see what happens after these gametes fuse. I could have labeled this dotted line as fertilization. We've got the fusion of 2 haploid cells and the formation of our first diploid cell. So we start really with the development of a zygote, our first diploid cell. And we're going to see how this first diploid cell that we call a zygote is going to grow and develop into what we call a blastula. Let me just describe this blastula real quick as a 64-celled hollow sphere. A 64-celled hollow sphere, and each one of those 64 cells is genetically identical. So we've got cell division without any cell differentiation. These cells don't start to develop into epithelial tissue, connective tissue, muscle tissue, or nervous tissue yet. They're all what we would consider true stem cells. This process in which we go from one diploid cell to 64 diploid cells without any cell differentiation is called cleavage. So cleavage is cell division without cell differentiation. Let's look at this in a couple of pictures. What's going to quickly happen after the blastula phase is we will start to see cell differentiation. The cells are going to differentiate into an internal layer of cells and an external layer of cells. That's when we get into this other term, that we call a gastrula. There will be cell differentiation. And then we see the cells differentiate into 3 layers, an endoderm, an ectoderm, and a middle mesoderm. We'll list which organ systems derive from those 3 different germ layers. And then we get into some functions of the placenta, stages of labor, and then a couple of differences in fetal circulation versus newborn circulation. Again, it's broad, it's a lot of topics. Then we're going to jump into heredity just so that I can introduce the terms allele versus gene, and then genotype versus phenotype, and we're going to see a couple of gene interactions. That's where I left a little bit of space. Let's see if we can get all the way through that. And again, stop me at any point. Yeah?
Is cell division without cell differentiation?
Yeah, cell growth without cell differentiation. The cells don't specialize. They stay true stem cells. Here is, we're going to see that picture, this process of cleavage and cell growth or cell division without cell differentiation. The first slide, I just want to point out where this process of fertilization occurs. That little dotted line that I drew on the board that shows sperm and egg meeting typically happens somewhere in the distal end of this fallopian tube or uterine tube. We can see sperm and egg, even though it's not a super detailed slide, we can see sperm and egg meeting in the fallopian tube. After fertilization occurs, the first cell, that first diploid cell we said was our zygote, it goes through this stage of cell division. Without those cells growing in size, those cells also are not differentiating yet. So we go through a few stages of, like we said, cleavage, cell dividing, cells not differentiating. Then we can't see a lot of the details here, so what's going to end up happening is an embryo is going to implant along this uterine wall. But let's jump to this stage, or this slide, to see that embryonic development. This is the blastula, that sphere of cells that are all genetically identical. We would consider these things true stem cells. They have all the information to develop into either epithelial tissue, nervous tissue, muscle tissue, or connective tissue. This is, we'll just look at this picture, it's a little bit cleaner. Here's our single zygote. When we go from one cell to a hollow sphere of cells, this 64-celled hollow sphere is our blastula. Then what we'll start to see is at one end of this blastula, a few of these cells are going to start to form an indention. And these cells that start to move inward are going to differentiate into an inner layer of cells and an outer layer of cells. Just a heads up, there's nothing new up here. These are reproductive handouts from last time. I'm just going through a few new slides of the pregnancy growth and development PowerPoint, and then we'll hit a little bit of heredity PowerPoint. The things we're pointing out here is zygote is our first diploid cell that forms after fertilization. We go from a zygote to a blastula, this hollow sphere of cells, by way of cleavage. Cell division without cell differentiation. Then this term gastrula refers to where the cells finally start to differentiate. At first, there are 2 cell layers that form this endoderm and then ectoderm. And in triploblastic organisms like us, we're going to see a 3rd layer. This middle mesoderm is going to form. So that red layer is the mesoderm, forming in between the endoderm in yellow and ectoderm in blue. So as that developing embryo continues to grow, it goes from that gastrula stage into the triploblastic. It's got 3 germ layers. The organ systems have not yet developed yet, but we can see these 3 germ layers kind of implant along this uterine wall, and they're going to start to grow these vascular tubes that develop in the placenta. Let's see what else. What I liked about this slide is it shows how we went from just 2 germ layer to that middle mesoderm layer starting to form in between the ectoderm and the endoderm. But the questions that we're likely to see about endoderm, ectoderm, and mesoderm is which organ systems come from these particular layers. Think about it this way, the stuff that's along the surface of your body, like a lot of nerve endings, if I'm feeling around in my pocket for keys or chapstick or whatever that is, money clip. I'm feeling around with nerve endings. So we have nerve endings on the surface of the body as well as skin. So ectoderm, the outer layers of tissue end up developing into things like the nervous system and the integument system. So skin, nervous system. The inner layer of cells, think the innermost organ systems in the center of the body, things like the digestive system, even part of the respiratory system. The exchange surfaces on the respiratory system are part of the endoderm layer. Mostly think digestive system and accessory structures to the digestive system form along the inside, the endoderm layer of cells. And then in between endoderm and ectoderm, the stuff in the middle is mostly muscular system, skeletal system. So the bones, the muscles, the blood vessels, that circulatory system. Remember, all of those blood vessels have smooth muscles through their walls. So all of the muscle tissue develops from this mesoderm. Okay, so we've gotten some examples of organ systems that are coming from each of those three different germ layers. The next thing that we'll list is the functions of the placenta. We started to see the formation of the placenta right here. These blood vessels that are going to start to deliver nutrients to the embryo and also help wash away the waste. When we look closely at this placental circulation, we're going to see that there's at least three things that are going on here. The placenta is not only delivering a lot of the fresh blood to the embryo, it's washing away the waste that's produced. The three functions are, the first thing that the placenta is going to convey to the embryo is hormones. We can see both peptide hormones, those protein-based hormones, and lipid-soluble hormones. So it serves as an endocrine function to the developing embryo. The second thing is kind of the first thing that I mentioned, the transfer of nutrients and washing away the waste. That also includes gas exchange that occurs right there between fetal blood and placental blood. Then the third and maybe not so obvious function, I think we mentioned it when we talk about immune system. We said there was a passive way of transferring antibodies from mother to newborn just by things like breast milk. Also through the placenta is another way that immune antibodies can be transferred. I think that might have also been mentioned in the slide, either by things like breast milk or through placental blood transfusion. So endocrine system, transfer of nutrients, washing away waste, and also transferring antibodies to the embryo that wouldn't have normally been there without the help of the mother. There's a lot going on. I just threw this slide in there that shows a comparison of the different organ systems and about how long these different organ systems take to complete development. The thing that I want to point out is the nervous system is what starts developing first and is actually the last thing to finish developing. So it takes a tremendous amount of time. And what's really going on there, we know in every other organ system, the nervous system controls all of these organ systems. So it can't finish developing until all of the organ systems have developed. It's got basically little axons kind of sitting in each of these organ systems. So it grows with the rest of the body. And I had this slide in there. What I ended up kind of not liking about this slide very much is it's hard to read some of those terms. But kind of as a review, there is the first diploid cell, that zygote. Then taking us from a zygote to this hollow sphere of cells, we said was the process of cleavage. That all happens before cell differentiation. So when cells start to differentiate, you know that you've entered this gastrula stage. The gastrula is when multiple layers started to form. And what I liked about this is that it shows, at least in humans, there are those 3 germ layers that are going to end up forming. And we have to know examples of organ systems that come from each of those germ layers. So that's what I kind of liked about this slide. We went through functions of the placenta. Let's list the 3 stages of labor real quick, and then we'll see the changes in fetal circulation. This is a chart similar to something we saw at the beginning of the semester, actually beginning of last semester, when we were talking about a comparison between negative feedback and positive feedback loops. This was a positive feedback loop where we said any kind of stretching along the uterine wall stimulated these glands of the posterior pituitary gland to release more oxytocin. Oxytocin is going to stimulate more of these uterine contractions, what's going to push that this baby now further down into the cervix, and as the walls of the cervix stretch even more oxytocin is released. So it leads to this positive feedback loop where we end up getting into the stages of labor. The first stage of labor is just when the cervix is going to dilate and thin out. So this dilation of the cervix is one of the first things that marks that labor is beginning. So as that cervix thins out, that's our first stage. The second stage is that the uterine contractions become so great that the infant is now delivered. So second stage is infant is delivered, and the third and final stage is that placenta is expelled. So that was nourishing the embryo as it was growing, and it's no longer there, so placenta is expelled in the third and final stage of labor. If we think about what's going on now with this infant, it used to get blood delivered that was already nutrient rich. It was already oxygen rich. It didn't have to get its own nutrients or oxygenate its own blood. But now, as soon as this newborn, this infant has made its way out, it has to start breathing air, oxygenating its own air. The circulation changes in a few places that are worth mentioning. So on the left-hand side, we've got the fetal circulation. The fetal circulation is still getting nutrient-rich and oxygen-rich blood from the placenta. If we look at a newborn on the right-hand side, obviously the placenta is not there, and there are a few changes along the blood vessels surrounding the heart that I want to point out. In fact, I don't think that this picture shows a close enough picture of the heart, so I added one more picture. And what I want to point out, let me make some room on the board. I want to point out really two structures. The two things that we want to point out, the very first top thing on the list is called the ductus arteriosus. This ductus arteriosus is going to connect the pulmonary, if I go to this slide, we can see that ductus arteriosus, this thing right here, it's going to connect the pulmonary trunk to the aorta. Right here, we call this thing the pulmonary trunk, and then you can see kind of right next to it is this aorta. In fetal circulation, what we can see right here, this pulmonary trunk, if we leave the right ventricle, we go through the pulmonary trunk, and that would take us out to the lungs if we were thinking about, newborn circulation. This pulmonary trunk branches into pulmonary arteries that went into either lung. But in fetal circulation, those lungs are not yet oxygenating blood. The blood that's coming into the heart is already oxygen-rich and nutrient-rich, so anything moving through the pulmonary trunk needs a way to bypass going into the lungs and can go instead straight into this aorta. So ductus arteriosus is going to connect the pulmonary trunk to the aorta. Again, the idea there is to bypass pulmonary circuit. Bypassing those lungs. The lungs don't have to oxygenate the blood. The other thing that we want to mention that is present in fetal circulation but not in newborn circulation is the foramen ovale. This foramen ovale, you can they've labeled it right there. This foramen ovale is a little opening that connects the right atrium to the left atrium. So it connects that right atrium, which is typically receiving oxygen, poor blood in newborn circulation or our circulation. But in fetal circulation, what's coming into that right atrium from the placenta is already blood that's nutrient-rich and oxygen-rich. So it can can go directly from the right atrium over to the left atrium and then be pumped all over the body. Again, the goal there is bypass lungs. We don't need to go from the right atrium down to the right ventricle and out into the lungs. The blood is already nutrient rich and oxygen rich, so it can just go straight to the left side and be pumped all over the body. There are a couple of other changes. There's a ductus venosus. Where's my pointer? Here's our ductus venosus that is bypassing the liver. Again, the liver doesn't have to store nutrients. The nutrients is already coming from the placenta. So there's a bypass through the liver. If we get into the heart, the ones we really need to mention around the heart, that ductus arteriosus is present in fetal circulation, just like this foramen ovale is present in fetal circulation. But after birth, we're going to see this ductus arteriosus closes up. It becomes a structure called the ligamentum arteriosum. You can still see it on those heart models. Now that I'm thinking about it, I don't think we had that on our lab list of things to see when we were looking at the heart, but you can still see number 10 right there is that ligamentum arteriosum. In fetal circulation, it was the ductus arteriosus. And then even if you look inside this right atrium, there's a little shallow depression. It's got a number 55 in it. That is the foramen ovale, and in fetal circulation, it was opened up and allowed blood to go from this right atrium over to the left atrium. Okay, putting the heart down, we've made it through our pregnancy, growth, and development introductory terms. We're going to switch gears real quick to look at some heredity. This is basic genetics, which is interesting. And all I really want to do here is separate terms like gene versus allele. There goes my markers. Is it okay if I erase this stuff? Pretty good with that? More terms. Just gene versus allele. I'll just define this real quick. A gene is going to basically be a segment of DNA. I've got to be a little more specific. I can't just say a segment of DNA. This is a segment of DNA that codes for a protein. It turns out when they sequence the human genome, of all of the DNA that is found in our cells, only about 2% of that DNA is actual genes. That means DNA that codes for a protein. Here's an example of codes for a protein. So maybe build an eye. So we all actually have a gene that says build 2 eyeballs. And I'm looking around, we all have 2 eyeballs. But the difference between a gene versus an allele, allele is basically an allele or alleles. These things are different versions of a gene. Versions of a gene. What is it?
Did it say build on I or an? I can't.
It says build an eye. Sorry. I probably could have spaced those out a little bit further. Yeah, build an eye. So we each have genes that say build an eye. But we might not all have the same version of a gene that says build an eye. So these different versions of a gene, maybe let me show some examples. There can be different versions of a gene. There's a version of a gene that says to build a brown eye. There's a version of a gene that says to build a blue eye. There are different versions of these genes, and they sometimes play together in different ways. Oftentimes there's a version of a gene that tends to completely mask the presence of other versions of a gene. So we'll see there's a dominant and recessive type of relationship between these versions of genes. Sometimes they blend together. So we'll talk about gene versus allele. We'll see there are different versions of a gene. Just like there's a gene that says to build hair. We have hair, but there are different versions of that gene. Build straight hair, build curly hair. Build wavy hair would be a mixture of the two. Let's just I know this is a very short video. It just has some interesting little moving parts to it. It will allow us to think about gene versus allele, and then we're going to, on the board, talk about gene interactions real quick. I'll just go to this picture and let this represent what I'm trying to draw or about to get to on the board, which is, we've talked about genes. Genes are little segments of DNA that build stuff. There are different versions of these genes, but I want to show where these genes are located. They basically ride on these structures called chromosomes. You can think of a chromosome as like a raft or like a boat that holds these genes on them. I'm thinking about that because, boats come in different sizes. A big boat is going to hold more people than a small boat. And a big chromosome is going to hold more genes on it than a small chromosome. Something like the Y chromosome is a very small chromosome, and it only has a handful of genes on it. But larger chromosomes, they're physically larger, and they can hold more genes along their length. What's interesting is that each different species of animal has its own unique chromosome number in the cells. So in every one of our cells, there are 46 chromosomes in each one of our cells. Each species has its own unique number. Chimpanzees is our closest living relative. They have 48 chromosomes in every one of their cells. Dogs have 76 chromosomes in each one of their cells. Some plants have hundreds of chromosomes in each of their cells. So it's crazy to think that there isn't anything about chromosome number per cell that has something to do with how advanced a species is. But anyway, each species has its own unique chromosome number. These chromosomes carry a bunch of genes along their length. And what's interesting about these chromosomes is you get a complete set of those chromosomes from your mom and your dad. These haploid numbers just indicate a complete set of chromosomes. I just said in human cells there are 46 chromosomes total. You get 23 of those chromosomes from your mother's egg and 23 of those chromosomes from your father's sperm. So if you think about it, there is, if we think of these chromosomes, to use an analogy like a book analogy. I'm looking around for a book and I don't have one. Imagine a book that's broken up into different chapters. Each chapter has pages of paragraphs, and in this analogy, each chromosome would be a chapter in that book. There is, in this case, in my analogy here, a novel on how to build a human. And that novel is 23 chapters long. You get a complete set of those 23 chapters from your dad, and you get a complete set of those 23 chapters from your mom. So you've got 2 copies of each chapter. These homologous chapters, the chapter number 1 from the dad and the chapter number 1 from the mom, these are what we consider homologous chromosomes. You got a chapter 2 from the dad and a chapter 2 from the mom, so on and so forth. 23 chapters, you've got copies from either parent. So what's helpful about that is that if you inherit a faulty copy of one of these instructions, you've got a backup from the other parent. Let's look at a couple of other vocabulary terms. We've separated gene from allele. So let me try and separate these other 2 terms, genotype from phenotype. We've already been using some of these things, and let me grab a couple of different colors. Genotype, you can really think of just as the collection of alleles. From either parent. So these collection of alleles, let's use this eye color as an example. I was just talking about brown eyes or blue eyes. There are different versions of these genes on how to build an eye. I'll let this uppercase B represent the version of a gene that says to build brown eyes. And I'll let this lowercase b represent the version of a gene that contains instructions to build a blue eye. So, and there are other versions. I've got just 2 listed here. It could be the case, and every one of our cells has two copies of this version of a gene for building an eye. It could be the case that if we look at our collection of alleles from both parents, we could have inherited, both copies of the allele that says build brown eyes. It could be the case that we inherited both copies of this recessive allele that says to build blue eyes. Or we could have inherited a copy of the brown allele and the blue allele. The terms that we would use to describe these different genotypes is, these are the same version of the allele. And it's the version of allele that is going to dominate the blue allele in this case. We would call this homozygous because they're the same, and dominant because it's the dominant allele. It's expressed even if there's just one version of it. So homozygous dominant. The next one is also homozygous because you've inherited the same version of this gene from either parent. And these versions of genes that you've inherited are the recessive versions. So homozygous recessive. And the last one here isn't homozygous at all. It's not the same. You've got different versions of the gene. So this type of genotype would be called heterozygous. So genotype falls under these three possible categories. It's the collection of alleles that you've inherited from either parent. You have either inherited the same versions of alleles from either parent. You either got the same dominant copies, the same recessive copies, or if you got a mix, we would refer to that as heterozygous. Phenotype, let me get a different color. I'm just going to draw a line here, and under here I'm going to try and define phenotype. Phenotype I'm going to describe as the outward display of a trait. So outward display of a trait. What this means, if we're thinking about our eye color example, the possible phenotypes are either going to be brown eyes. We'll separate those a little further. Brown eyes or blue eyes. Or depending on the combination of alleles, it could be a mixture of the two. It could be something totally different. Phenotype is what you see. So it's this outward display of the trait. These outward displays of traits are basically determined based off of the genotype. In our gene interactions that we've introduced so far, if you have just one of these dominant alleles, so this one as well as heterozygous, both of these are going to result in an individual that would have brown eyes. And there's only one possibility here. I guess I'm losing all of my colors. Only one possibility, if you have both of those recessive versions, would you end up getting blue eyes. So there's two possible phenotypes, brown eyes or blue eyes, the stuff that we see. And these possible phenotypes are they're determined based off of the genotypes. So genotypes are the collection of alleles. As you can kind of start to see, depending on how these alleles are going to play together, sometimes just one allele is all that you need to display a trait, like in the case of brown eyes. But in the case of blue eyes, in order to display blue eyes, you need two copies of that allele. That's where we get this term recessive versus dominant. And I should also say that recessive doesn't mean bad. Dominant doesn't mean good. There are some things like dwarfism is something that you probably wouldn't want to have, but it's a dominant trait. If you have just one of those alleles, that's something that can lead to dwarfism. Again, recessive just means that it takes two of those alleles to display that trait. Dominant just means it takes one. Okay, let's catch back up with some of these slides. Like we can now see on the genotype side, the genotype is showing us the collection of alleles. Either heterozygous in the middle, or homozygous on top or bottom. Homozygous recessive down there and homozygous dominant on top. From this type of gene interaction, you can see once again it's a dominant and recessive type of interaction, because as long as you have one of those dominant alleles, you display that dominant trait. The only way that you can display the recessive trait is to have two of those recessive alleles. Don't think recessive bad or dominant good. But this isn't the only type of gene interaction. It's a common type of gene interaction. You can see it's still the type of interaction that we're looking at here. In the case of these birds, it looks like red feathers is the dominant type of allele. So you could have red feathers with two versions of that gene or just one version of the gene. In order to display the blue feathers, you'd have to have two of those recessive alleles. Okay, so that's fine. Let's look down here at this picture. This is what I kind of like this bottom part of the graph, because it's reminding us of homologous chromosomes. These are representing homologous chapters, let's say chapter 1 from the dad and chapter 1 from the mom. If we went through and read those chapters, each chapter would have its own paragraph, and chapter 1 from the dad is going to include the same genes that you would find on this homologous chromosome from the mom. If this gene is for building eyes, then you're going to have instructions for building eyes on the chromosome 1 from the mom and instructions for building eyes on chromosome 1 from the dad. What these two different colors are showing us right here is that the instructions that you've inherited from your dad for building eyes are not the same instructions that you've inherited from your mom. So this is showing us examples of, this would be a genotype that we would consider heterozygous, different copies of the allele. But for gene B, we can see that both chapter 1 from the dad and chapter 1 from the mom have the same version of gene B. In gene C, we can see different versions of the gene. So this is going to be heterozygous. Are those genes going to play together in a dominant recessive type of interaction? Are they going to blend together? Gene D, once again, is homozygous. We don't know if that's dominant or recessive. But gene E, once again, is heterozygous. So homologous chromosomes have the same genes along their length. They may be different versions of those genes if we look close. So at least 3 places we're showing heterozygous combinations of alleles. And in 3 other places we're showing homozygous combinations of alleles. Homozygous the same, heterozygous just different copies of alleles. Okay. Last thing that we're going to add to the genetics component is that there are different versions of genes. We call those different versions of genes alleles, and they don't always play together in this dominant recessive type of relationship that we've been talking about. Again, dominant, you just need one version of those genes for that trait to be displayed. In this case, red flowers is dominant. You just need one version of red flowers and you get solid red flowers. The only way to get white flowers is to have 2 of those copies of instructions that say build white flowers. But then when we look down here, we've got flowers that are not red, they're not white, they're, in some cases, some flowers are both red and white. This is going to be codominant, versus these flowers that are neither red nor white. They are a blend of the two. These are pink flowers, obviously. I want to make a distinction between what we're going to call incomplete dominance and codominance. Both of them, they sound like they could be the same, but to make a distinction, in codominance, both alleles are dominant. Both are displayed. So if we think about red and white alleles, in a codominant type of relationship, you'll get some part of the flower that's completely white, just like it would be if it was the only set of instructions. And you get other parts of the flower that are completely red, like they would be if it was the only instructions present. So in codominance, we can see both alleles are expressed in their full, as if they were the only ones there. But in incomplete dominance, you have a blending of the 2 alleles. Right? Nowhere on this flower are there just red petals like there would be if it was the only instructions, or nowhere on there would there be white petals, but instead you see a blending of the red and the white. You get this pink petal.
So in humans, would it be like one brown eye, one blue eye, or is that?
An example in humans of incomplete dominance would be this one, hair.
The wavy hair.
the wavy hair
If it's
versus
codominant, would that be, what would it,
going back to this one in what?
codominant?
Oh, so if you're thinking about the blue eye and brown eye, what's happening there is if we go all the way back, I'll see if I can do this without going all the way back in the slides. When we were going to that embryonic development slide, and we were seeing that at one point 3 different tissue layers started to develop, and from one of those tissue layers, the nervous system started to develop. So if we think about all of the cells that are differentiating into ectoderm cells, right? There's thousands of these cells that are starting to divide and become different parts of the nervous system. And at one point, these cells are all genetically identical and they all contain instructions for building brown eyes. All that's happening, it's what's called a somatic mutation. What I was about to try and show here is that every one of my cells has instructions for building blue eyes and brown eyes. And if you're heterozygous like this, then every cell just has one instruction to build those brown eyes. And in this process of somatic mutations where each of those ectoderm cells are differentiating, at some point they start to differentiate into a left eye and a right eye. And after these 2 cell lineages differentiate, if there's a mutation to one of those lines of cells where this one instruction to build brown eyes gets cut off due to a mutation, now that entire lineage of cells is going to lose pigment. So that eye is going to be completely blue. And in the other lineage of cells, if the mutation happened after those 2 cell lineages diverged, then one eye is still going to have a functioning brown allele to produce brown pigment. And the other eye, just in the divergence of those 2 cell lineages, that somatic mutation happened after the divergence, so one eye has lost its pigment. So you're really seeing the loss of kind of the turning off of a gene in one line of cells. Yeah.
Just to verify, because I do know a person who has like a brown eye, like a pretty brown eye and a blue eye.
Yep.
And he's a little bit lighter than me, but, so that's a somatic?
That would be a somatic mutation. And that happens all the time, like, in breast cancer, you get cancer on one breast and not the other. That's a somatic mutation. It happened in one lineage of tissues and not the other. So I got off topic there a little bit with that question, but so your first thing was about the brown eye versus the blue eye. So that would be an example of one of those somatic mutations, which can happen in a bunch of different examples, like I mentioned with cancer. The second part of that question was to give an example of codominance in humans, which is where we're going. In fact, we're going to see examples of each of these interactions in humans. And the first interaction is the one that we were mentioning, the fact that there are different versions of genes that have instructions to build an eyeball. This is an example of the Punnett square that I was about to draw, where you can have an individual that has brown eyes. So this would be the phenotype, the output, brown eyes. And this brown eye is determined based off of the genotype. In this case, each parent, we can say this is maybe the male and up here would be the female. In this case, both parents are heterozygous for having brown eyes. And as long as they have one set of instructions for building brown eyes, the eyes are going to end up brown. And being both heterozygous, at least 25% of their offspring are going to inherit 2 of these recessive copies, 2 instructions to build blue eyes. So about 25% of the offspring from 2 brown-eyed heterozygous individuals are going to be blue eyes. Again, not probably easily explainable 60 years ago. It would be probably wondering who the milkman is or something like they're the FedEx man nowadays. But then if we cross, this would be a blue-eyed individual with a brown-eyed individual. This would be an example of my mom that has blue eyes and my dad that has brown eyes. Turns out, background information on my dad, his mom had blue eyes, my grandmother. So my dad has brown eyes, but he's heterozygous for brown eyes. Half of his gametes are sending instructions for brown eyes, and the other half of the gametes are sending instructions for blue eyes. That's how I inherited my brown eyes, and my brother, my only other sibling, has blue eyes. Anyway, this is a dominant recessive type of interaction. You only need one of those versions of genes to display that trait. Here are a list of other types of dominant and recessive traits. I mentioned whether the earlobes are attached or kind of detached. Freckles are something that are dominant versus recessive. Hair color, whether you have dimples or a widow's peak or a cleft chin, all types of dominant recessive interactions. But that's not the only type of interaction. Maybe the next one that we need to mention is this codominance. These are all flowers displaying this codominance. We can see in some of the parts of the petals just white petals, and other parts red petals. There's no blending of the two. But this isn't a plant class. This is a human A and P class. So our human example of codominance is going to take us back to blood types. When we were talking about blood types, we said on the surface of your red blood cells, if you have type A blood, you have little A's, what are equivalent to little A's on the surface of your red blood cell. And if you have type B blood, you've got little B's on the surface of your red blood cell. And if you had type O blood, you didn't have any of those surface markers whatsoever. But people that had both A and B alleles, if we could look closely at the surface of their red blood cells, we would see codominance. We would see tiny little A's in the shape of an A, just like it would be if it was the only protein on the surface. So those A's look just like the A's on type A blood. And the B's that show up on the surface of that red blood cell are identical to the B's on type B blood when it was the only allele present. So in codominant, you've got both alleles are fully expressed. We've got true A's and we've got true B's. We don't have, for example, if this was incomplete dominance, this wouldn't look like an A or it wouldn't look like a B. It would look like half A, half B. It would be a blend of the two. Let me know if that is close enough. Like if we think of these A's as being red petals and the B's as being white petals, on this we would see both red petals and white petals. It would look something like this, where there's red petals and white petals. If instead this was an incomplete dominance, we would see, instead of red petals and white petals, we would see every one of these would be a pink petal. Wouldn't be red, wouldn't be white, it would be something in between. So in blood type, we don't see that. We see true A's just like they would be all by themselves, and true B's just like they would be all by themselves. So this would be codominance in humans. And incomplete dominance in humans. Again, incomplete dominance, there's a blending effect. So this is supposed to be a red flower, even though it looks kind of purple. And this is supposed to be, well, this is a white flower. You can see the cross between the two. We don't get red or white flowers, we get pink flowers, something in between. In humans, the example here would be wavy hair. So straight hair that's one allele for hair type. Then curly hair is another allele for hair type. These two alleles play together in a way in which they blend. So a cross between these two, you get kind of a blending of the straight hair versus the curly hair in what we would consider wavy hair. We don't have to draw Punnett squares in here. I won't hit you with genotypic or phenotypic ratios, which is something we would do in a genetics class. I just simply wanted to separate the terms genotype versus phenotype. I wanted to show that not all genes play together the same way. Sometimes, in fact, the most common type of relationship is this dominant recessive type of relationship, but there are other types like codominance or incomplete dominance.